Micro folks, this is our continuation of our introduction to aerobic respiration, metabolism, PowerPoint B. We had just described how crucial the evolution of porphyrin rings were to the evolution of aerobic environment, of aerobic respiration. So not to panic, you guys, you don't have to memorize what these porphyrin rings look like. But we do want to remember that porphyrin rings were crucial for evolution of bacterial chlorophyll, and with minor changes that permitted evolution of chlorophyll A and oxygenic photosynthesis. And then porphyrin rings are essential part of heme prosthetic groups, and these heme prosthetic groups are important components of cytochromes, which are crucial for the construction of electron transport chains. So these porphyrin rings really were the big event when we describe how aerobic respiration evolved. So this is the big overview, folks, before we get lost in all the details. So this is, you might remember, folks, we said for lecture exam 2, for one of the short answer questions, I'd ask you to write this redox reaction summarizing complete oxidation of glucose via aerobic respiration. So remember, these are our reactants. Our glucose is our source of high energy electrons. Oxygen will be the terminal, the final electron acceptor at the end of the electron transport chain. This arrow represents the next 2 hours of lecture. We want to remember that in this multi-step process, slowly electrons are going to be stripped from what used to be glucose. So slowly glucose is oxidized. So all the 6 carbons present in glucose eventually are going to get blown off, released as 6 carbon dioxide. So glucose is oxidized to carbon dioxide. At the end of the electron transport chain, when all these high energy electrons have given up their energy so that the ETC can pump protons across the gradient, across the membrane, those tired out electrons, remember, they can't be just thrown into the cytoplasm. They cause a lot of damage. So in aerobic respiration, our terminal, our final electron acceptor will be molecular oxygen. So in this example, then, it's the molecular oxygen is going to be reduced to water. And why is the cell doing this? Is to release a huge amount of energy. About a third of it will be captured in making ATP. And these are the hypothetical maximum yields, 36 to 38 ATP, depending on whether you're a prokaryote or eukaryote. And then 2/3 of the energy will be lost as heat. So key events in aerobic respiration, it's the complete oxidation of glucose, not partial oxidation as we saw in fermentation. Here we have electron transport chain that's going to use high energy electrons from those organic molecules to create a proton gradient across a membrane. It requires oxygen in an aerobic environment. Molecular oxygen acts as a terminal electron acceptor. This is more accurate range of ATP production. And later we're going to see, folks, that the ATP is produced using a combination of substrate level phosphorylation and oxidative phosphorylation. And you might recall oxidative phosphorylation is going to use chemiosmosis, the proton gradient, to help drive cellular work ATP synthesis. Our end products are going to be far less toxic than the alcohols and acids of fermentation. We're going to get up with relatively non-toxic CO2 and water. And another cool thing, folks, that we won't have much time to discuss is the intermediates of aerobic respiration. Many of them are going to be used to help drive synthesis of other organic molecules, such as nucleotides and amino acids and lipids. So always good to know where cellular respiration occurs. So we're going to focus, folks, on bacteria. So we're going to be focused on prokaryotic cells. So we'll see there's numerous stages in aerobic respiration. So the first 3 steps in aerobic respiration in bacteria occur in the cytoplasm. Okay, so that will be glycolysis. An intermediate step we're going to call the Krebs Prep and the Krebs cycle. All those enzymes are in the cytoplasm. And then the big events, the electron transport chain and ATP synthase, the electron transport chain and ATP synthase are going to be located in the cytoplasmic or cell membrane. In contrast, in eukaryotic cells such as animal cells, glycolysis will occur in the cytoplasm, but then the remaining steps are going to occur in the mitochondrion. So the so-called Krebs Prep, the Krebs cycle, will occur in the mitochondrion. And the electron transport chain and ATP synthase will be located in the inner mitochondrial membrane. And this is so neat, you guys, because according to the endosymbiotic theory, mitochondria evolved from primitive aerobic respiring bacteria. And according to the endosymbiotic theory, then the inner mitochondrial membrane evolved from what? The primitive bacterial cell membrane, right? The ETC is located there, and ATP synthase is located there. So again, folks, just comparing and contrasting fermentation, relatively simple, but low ATP yield, like 2 ATP per glucose, high energy waste products and toxic waste products. We can see in aerobic respiration, it's a much more complicated process, but the advantages are we're going to completely oxidize glucose. We're going to use all those high energy electrons to drive approximately 36 to 38 ATP. Our end products are going to be far less toxic, carbon dioxide and water. So again, there's many steps, many parts of aerobic respiration. So just the overview, we'll talk about glycolysis, the Krebs Prep or preparation or production of acetyl-CoA, the Krebs cycle. We'll then take a look at how the electrons that have been ripped off of glucose will be delivered to the electron transport chain, and the ETC will use those high energy electrons for active transport to pump protons across the cell membrane to form the proton gradient. And then we'll see how the proton gradient will drive the chemiosmotic production of ATP via ATP synthase. So glycolysis, folks, is going to be just as we saw it in our introduction to fermentation. So again, we'll start out with the glucose. We'll have partial oxidation of the glucose. We're going to transfer a total of 4 high energy electrons to NADH. We're going to end up with 2 reduced NADH. We'll make a little bit of ATP by substrate level phosphorylation. But you guys, this is the big event here. This pyruvic acid, 2 pyruvic acid per glucose, still chock full of high energy electrons. So the next steps will be to finish stripping off those high energy electrons, to finish oxidizing what used to be glucose. And furthermore, during that process, we're going to totally rip apart the carbon skeleton, releasing all of the carbons, the 6 carbons of glucose as CO2. So this next step, folks, we're going to call it links glycolysis to this Krebs cycle. I'm going to call this the Krebs Prep, or the production of acetyl-CoA. So let's take a look at this linking step right here. So this would be stage 2, the Krebs Prep. And here's our pyruvic acid, folks, the end product of glycolysis. And you'll see there's some really neat things happening in this Krebs Prep. We're going to start to further tear apart the carbon skeleton. We're going to rip off this 3rd carbon and release it as carbon dioxide. So this process is called decarboxylation. Then we're going to rip off some more high energy electrons from what used to be glucose, and we're going to pass them to our NAD to make our reduced NADH. And then this little 2 carbon residual, what used to be glucose, still carrying high energy electrons, we're going to link it to a carrier molecule called coenzyme A. So at the very end then of the Krebs Prep, we have this cool molecule called acetyl-CoA, the acetyl group, still carrying carbons and electrons from glucose. We've also produced reduced NADH, and we've also produced carbon dioxide. The 3rd stage is when we finally finish oxidizing what used to be glucose. We're going to rip off all the remaining high energy electrons, and we're going to finish tearing apart the carbon skeleton, releasing it as the carbons will be released as carbon dioxide. So this is called the Krebs Prep. This is called the Krebs cycle, also known as the citric acid cycle, also known as the tricarboxylic acid cycle or TCA cycle. On a short answer, you could use any of these terms for it. Let's just tear it apart first. So we'll see we're going to introduce the acetyl group, the 2 carbons and the electrons that used to be part of glucose into the cycle. We're going to combine those 2 carbons with this 4 carbon acceptor molecule, oxaloacetic acid. So if we take 2 carbons and 4 carbons, that's going to make this first intermediate called citric acid. And even though we can't see it here, folks, citric acid is really unusual. It has 3 carboxyl groups on it. Rather unusual, and that's one reason this is called the tricarboxylic acid cycle here. Okay. Also, because the first intermediate is called citric acid, this is one reason it's called the citric acid cycle. Now, you don't need to memorize individual enzymes nor individual steps, but we do want to know what are the key events here. So if we go around the cycle once quickly, folks, we'll see Here we're going to rip off more high energy electrons, pass them to NAD to make reduced NADH. We're going to rip off carbons. Here we're going to rip off another carbon, release it as carbon dioxide. So again, just as we saw in Krebs Prep, this is called an oxidative decarboxylation. In the next step, we're going to have another oxidative decarboxylation. We're going to make another NADH. We're going to rip off more carbon and release it as CO2. Here, and this isn't that important, folks, but here we get that a carrier molecule coenzyme A getting involved. Don't need to worry about this, folks, but we're going to have phosphorylation of the intermediate. That phosphate group, ultimately, it's going to take a couple of steps, will get donated to ADP to make ATP. So we're going to get a little bit of ATP production by substrate level phosphorylation. Then in the final steps of the Krebs cycle, we're going to finish ripping off all the remaining high energy electrons that once used to be part of glucose. Now here, folks, we see the second electron hydrogen atom carrier we had talked about earlier. This is FAD. When it's reduced, it's FADH2. Just remember that the electrons carried by FADH2 are a little bit lower energy than the electrons carried by NADH. And again, that will become important when we start adding up how many ATP we're going to make. The reason this is called a cycle is that we end up regenerating the original acceptor molecule, this oxaloacetic acid. This is what you need to know for the exam. So per one acetyl CoA that enters the cycle, we're going to get 2 carbon dioxide, 3 NADH, 1 FADH2, 1 ATP indirectly via substrate level phosphorylation, and 1 oxaloacetic acid will be regenerated. We just have to remember, folks, that per glucose, we're going to end up with 2 acetyl CoA. So if I ask you for the number of end products of the Krebs cycle, starting with 1 glucose, you just need to remember you have to double everything. So 4 CO2, 6 NADH, 2 FADH2, 2 ATP, and we'll regenerate 2 OAA. And again, this was just to remind me to share with you the reason it's called a cycle is that we're going to end up regenerating that original oxaloacetic acid. Okay, so at this step, we still haven't made much ATP. We've ripped off all the high energy electrons from glucose, which are now being carried by NADH and FADH2. So we could ask, so where's all the ATP, right? So you might recall, folks, that most of the ATP is going to be made by this cool new process, oxidative phosphorylation, that involves the electron transport chain, the proton gradients, and ATP synthase. And again, just a big overview here, folks. So high energy electrons that we've stripped off of our glucose or other organic molecules are going to be delivered to the electron transport chain. They'll be passed through the electron transport chain, and with each passage, each transfer of electrons from one member of the chain to the next, a little bit of energy is released. And we will say that some of that energy released from the electrons can be used by the proton pumps of the electron transport chain to pump those protons across the membrane. So again, folks, key things to remember: the function of the electron transport chain is to pump protons across the membrane. Very important, folks, what's the energy that drives electron transport chain function, proton pumping? It's a high energy electrons from glucose or other organic molecules. Once the proton gradient is formed, then it's the diffusion of protons from an area of high through ATP synthase to an area of low proton concentration. It's this flow of protons that's going to drive ATP synthase. It will assist ATP synthase to phosphorylate ADP with phosphate to make our ATP. So the energy source for ATP synthase is the proton gradient. I think what we'll do here, folks, maybe we'll stop here just trying to break this up a little bit. Oh, just so remember, folks, chemiosmosis is when we use these chemical concentration gradients across membranes to do work. So ATP synthesis is an example of cellular work, biosynthesis, right? And so it's this chemiosmosis then that helps drive ATP synthesis. So we could call it chemiosmotic production of ATP if we wanted to be fancy. This is a cool exam question, you guys. So if our ETC is functioning properly, which side of the membrane would have the lowest pH. And just remember that inverse relationship between pH and hydrogen ion concentration. So this side of the membrane would have the lowest pH, right, the highest hydrogen ion concentration. This side would have the higher pH, the lower hydrogen ion concentration. Okay, I think folks will stop here. These next slides, they're so text-rich, probably not great slides, but it's just to kind of walk you through the process one step at a time of how the ETC works, how the proton gradient is going to drive ATP synthesis. So we'll stop here and hopefully we'll get another audio movie in describing these events.